Good morning, second years. Today we are going to know about the Unit-5 embryology. In embryology, the first topic is the gametogenesis. We all know that gametogenesis is the process of division of the diploid cells, the two end cells, to produce the new haploid cells. In humans, there are two types of gametes are present. They are male gametes and female gametes. Male gametes are called as sperms and the female gametes are called as ovum. Next, in the spermatogenesis, this is the testis and this is the epidermis region. In this, number of seminiferous tubules are present. In each tubules, there sperms are here. The sperms will be formed. This is the cross sectional of the seminiferous tubules. This is round is called as spermatogonium. So first, the primordial germ cells. The PMCs divide into the spermatogonium. After the mitotic division, this is called as spermatogonium. After the mitotic division, it is again divided into primary spermatocytes. This is called as primary spermatocytes. After the meiotic division, it is again divided into secondary spermatocyte. These are called as the secondary spermatocytes. After this stage, this secondary spermatocytes again divided by the meiotic division to the secondary spermatocytes is divided into spermatids. These round structures are called as spermatids. Finally, these spermatids develop into a mature sperms. This is the lumen of the seminiferous tubules. This is how the sperms are produced in the seminiferous tubules of a male reproductive system called testis. This is the structure of sperm. This is the acrosome. This entire part is called as the head region. This is the middle piece region, and this one is called as the tail. This is the acrosome which helpful to break the outer layer of the ovum, and this is the head region. In head region, we can observe nucleus is present. Under the nucleus, there is a centriole is present. This is the attachment between the middle piece and the head is called as the neck region. In the middle piece, we can observe this is the we can observe the mitochondria, which is the energy source for the sperm, and this is the tail region also called as flagellum, and this is called as the plasma membrane. This is the structure of the sperm. Next, oogenesis. Likewise, in spermatogenesis, in oogenesis, ovum also produced in some of steps. In oogenesis, first the primordial germ cells, it develops into the oogonia, and next the oogonia enlarges and develops into a primary oocyte. And again, the primary oocytes, after the first meiotic division, it divides into secondary oocyte and the first polar body. Next, the secondary oocytes again, after second meiotic division, it is divided into ovum and a second polar body. The first polar body is here, and this is the second polar body. This develops into a mature oocyte. This is the structure of ovum. This is the corona radiata. This is the zona pellucida. This is the vitelline membrane, and this is the nucleus, and this is the gemule vesicle, and this is the ooplasm. And next, topic, fertilization. We all know that fertilization is the process in which the male and female gametes are fused together, and this process initiates the development of a new organism. The male gamete is called as the sperm and the female gamete is called as the egg or ovum. These are the specialized sex cells which fuse together, and to begin the formation, it fuses and develops a formation called a zygote, which is this entire process is called as sexual reproduction. In the sexual reproduction, we can observe four steps. That is the approach of the spermatozoon to the egg. The process, the spermatozoon, with the help of the tail, it moves to the near to the egg, and next penetration of the sperm. Next, the reaction in egg during the sperm penetration and the behavior of sperm within the egg. Penetration of the sperm is done with the help of the acrosome. The acrosome helps to break the wall of the egg, and the reaction in egg during the sperm penetration, when the sperm is getting penetrated into the egg, the reactions happen in the egg. Some of the reactions will happen in the egg, and the behavior of sperm within the ovum. When sperm enter into break the all walls of the egg, the nucleus present in the sperm body enters into the ovum ooplasm. Here, sperm make contact with the egg. Here, some of the hydrolytic enzymes are getting secreted into the zona pellucida. This is the sperm getting attacked. This while attaching to the zona pellucida wall of the egg. Next, acrosome reacts with the pellucida. This is the acrosome, the first layer of the sperm. It reacts with the zona pellucida. Here the hydrolytic enzymes are secreting into the zona pellucida. Next, acrosome reacts with the perivitelline space. These acrosome enters into the, this is the perivitelline space, enters into the perivitelline space. Here these are the proton receptors which are present in the egg. Next, plasma membrane of sperm and egg fuse. The plasma membrane, this is the plasma membrane of sperm and the egg. The attachment is happening. Next, sperm nucleus enters egg. Here the sperm nucleus is getting entered into the egg. The cortical granules fuse with egg plasma granules, which renders the vitelline layer impenetrable to sperm. The granules which are present in the sperm fuse with the egg plasma membrane. It helps to renders the vitelline layer, which is impermeable to other sperms. Next, the types of eggs which are we can observe in animals. Ovum is the female gamete. It stores the food required for the entire process of development in the form of yolk. The functions of this egg are, it supplies a haploid set of chromosomes to the future embryo. It also contributes almost all the cytoplasm of the zygote. It supplies food to the developing embryo. Based on the distribution of yolk, it is divided into 4 types: isolesthal, telolesthal, centrolesthal, and discoidal lesthal. Next, the classification of egg. Based on the amount of yolk, here we have observed the distribution, how the yolk is distribution. It is divided into isolesthal, telolesthal, centrolesthal, and mosaic-lesthal, which means the yolk is distributed sidewise, center, and discoidal. Here, how much the amount of yolk is present, it is divided into 4 types: isolesthal, which means the cells with small and eventually distributed yolk. Next, microlesthal, mesolesthal, and macrolesthal. These are the types of eggs which are based on the amount of the yolk. This is the cleavage. What is cleavage? A cleavage is a rapid cell division that leads to a multicellular embryo. First, when the male gametes and the female gametes get fused, after that a zygote will be formed. Again, first the zygote undergo cleavages. It got divided into some of the pieces. After 2 cells, 4 cell, and 8 cell stage, this is the 8 cell stage. After this cleavage, a group of cells called as blastula is formed. After that, if we cut the blastula into the 2 pieces, a hollow space is present inside the blastula is called as the blastocele. This is the cross section of the blastula. Again, the blastula undergone a process called gastrulation. In this, the gastrulation, the outer layer invaginates into the inner layer of the egg. This process is called the gastrulation. After this invaginates into the internal layer, it develops into three layers: ectoderm, endoderm, and the mesoderm. Here this is the blastocele. Here the entire hollow space is getting reduced when the gastrulation process is going on. This is the blastula, this is the endoderm, this is the ectoderm, and this is the mesoderm. This the entry of the pore is called as the blastopore, and this entire is called as the gastrula. Next, there are two types of cleavages called indeterminate cleavage and determinate cleavage. In indeterminate cleavage, if these cells are separated from each other, each has the potential to develop into an entire organism on its own. But in determinate cleavage, if these cells are separated from each other, they will die. They do not have any potential to develop into a new organism on its own. And holoblastic cleavage, if the division is divided like this, first 2 cell stage, 3 or 4 cell stage, and it is a parallel division we can observe. This type of division is called as, if the cell is divided entirely, is called as a holoblastic cleavage. If we can observe that in this cleavage, in meroblastic cleavage, in this type of cell, only one side of the part is getting cleavaged, getting divided on one side only, not entirely. Here in holoblastic, the entire cell is getting divided, but in meroblastic, only some part of the cell is getting divided, is called as meroblastic. Next, the development of frog up to the formation of primary germ layers. This entire process is done in the 3 steps called as fertilization, cleavage, and gastrulation. We all know that fertilization, this occurs externally in frogs. Typically in water, sperm fertilizes the egg, forming a zygote. In frogs, the reproduction will be done externally in frogs. And next, cleavage. The zygote undergoes a rapid cell division, forming a solid mass of cells called as morula, which then develops into a hollow ball of cells called as blastula. In the previous slide, we studied the blastula, gastrula. Likewise, after the rapid cell division of zygote, it develops into the morula, and then it develops into the blastula. Gastrulation involves the inward migration of cells to form 3 germ layers. In the previous slide, we have told that gastrulation, which means the inward growth of the cells, it develops a 3 germ layers called ectoderm, endoderm, and mesoderm. Ectoderm, the cells on the surface of the blastula give rise to the outer layer of the embryo, which will form structures like the skin and nervous system. This outer layer of the blastula develops skin and nervous system. And next, this mesoderm migrate between the ectoderm and the endoderm, form the middle layer of the embryo. This layer gives rise to the structures called muscles, bones, and the circulatory system. And in endoderm, these cells migrate into the innermost. This is the innermost part of the blastula. Here the endoderm develops into the digestive system, respiratory system, and the internal organs.